Hey guys, welcome back to my YouTube channel, and today I will be covering Unit 4 Enzymes. Just like before, let's go with the syllabus guide first so that you guys would have an idea about what will be covered. If you think that this is necessary, you may pause the video and take a quick screenshot and self-check at the end of the video to see if all the parts have been covered. Right. Before actually talking about enzymes, first we must be familiar with the term catalyst. As per the direct definition, a catalyst is a substance that speeds up chemical reaction by providing a path with lower activation energy. And an important characteristic of a catalyst is that it remains unchanged at the end of the reaction and can be used again. This part you guys might have learned for chemistry as well, but I will quickly explain that with the diagram as well. As you can see, the two diagrams on the top are the energy profile diagrams for regular endothermic and exothermic reactions. Hope you are familiar with these two terms, but if you're not, don't worry much about it because all you have to understand is about the activation energy and the role played by a catalyst. Activation energy, as indicated in the diagrams by the blue arrows, is the minimum energy requirement for a chemical reaction to take place successfully. For endothermic reactions, the activation energy is quite high when compared to exothermic reactions. Now, when we add a catalyst to the same reaction, the reaction will speed up. The logic behind it is that the catalyst reduces activation energy required and provides another pathway for the reaction to take place, which is represented in the two diagrams below for both of the types of reactions. The reduced activation energy is represented in the diagram with another curve that goes below the initial one, which is drawn in red in the diagrams. For exothermic one as well as the endothermic one, when a catalyst is added, the reaction can happen with lesser activation energy, meaning that the reaction will become faster. Hope you all understood that. So with that in mind, enzymes are referred to as biological catalysts that speed up metabolic reactions that happens in living organisms and remains unchanged at the end of the reaction. There are two main types of enzymes, intracellular and extracellular. Intracellular enzymes are those that function inside cells, whereas extracellular enzymes are those which are let outside to function or do its part. Now let's talk about some of the properties of enzymes. All the enzymes are protein molecules, but not all protein molecules are enzymes. That is the most important thing. All enzymes are protein molecules, but not all proteins are enzymes. Secondly, these enzymes are made in the cytoplasm under the instruction from genes which are found in the chromosomes of the nucleus. Thirdly, all enzymes work best at a particular temperature and a pH value known as the optimum temperature and optimum pH respectively. And generally, the optimum temperature for most enzymes would be 37 degrees Celsius, which is the normal body temperature, and the optimum pH would be 7 or neutral conditions. Finally, remember that enzymes are specific, meaning that each enzyme can catalyze only one kind of reaction. Now it's the most important part of this lesson, which is the lock and key mechanism, or basically how enzymes work. First, let's get familiar with the key terms. Substrate is the thing that binds with the enzyme, also referred to as the key, whereas enzymes is referred to as the lock. So each and every enzyme has an active site with a specific shape that only allows a specific substrate to fit in, similar to the concept of each lock having a specific key. Then the enzyme and the substrate would bind or connect at the active site like a key fitting a lock, shown in the second diagram here, and forming the enzyme substrate complex. According to the diagram here, the initial substrate is broken down into two products and the products leave the active site, making the enzyme available to be used again for the same reaction. It must be noted that enzymes would either combine two substrates at the active site forming a single product, or would either split up a single substrate into two or more products. These reactions that split larger molecules into smaller molecules are known as catabolic reactions, whereas reactions that combine smaller molecules forming larger products are called anabolic reactions. Also note that this lock and key hypothesis is also seen in the digestive system and is referred to as hydrolysis. And the names of enzymes end with ASE and are named according to the substance it acts upon. Those last 3 points are some additional information for y'all. So remember that catabolic reactions split larger molecules into smaller ones, and anabolic reactions join smaller molecules to form a larger one. Also remember that when measuring the progress of an enzyme catalyzed reaction or the enzyme activity, we can always either measure the concentration of reactants or the concentration of products. If the concentration of reactants are measured and a graph is plotted, it will be a descending graph as the concentration of reactants gradually decrease with the reaction time. If the concentration of products are measured, it will be an increasing graph as the concentration of products gradually increase as the reaction progresses with time. Now I have given some basic examples of digestive enzymes to prove to you all that almost all the enzymes end with ASE. As you can see, except for pepsine and trypsin, all the other enzymes end with ASE. And under starch digestion, we identify 3 enzymes: salivary amylase, pancreatic amylase, and maltase, whose final product would be to form maltose after breaking down carbohydrates. In protein digestion, we are talking about 3 enzymes: pepsine, trypsin, and peptides, whose goal is to break down large protein molecules into peptides and amino acids. In nucleic acid digestion, we have nuclease and nucleosides. And for finally fat digestion, we have lipase, whose final goal is to form fatty acids and glycerol after breaking down lipids. Then we move on to the 2 main conditions that affect the activity of enzymes, which are temperature and pH. First, let's talk about the effects of temperature on enzymes. Generally, the optimum temperature for most enzymes is 37 degrees Celsius, which is the human body temperature. Generally, when the temperature is increased, kinetic energy of molecules increase, and the enzyme activity therefore increases, because the frequency of successful collisions between molecules would increase. But when the temperature is increased beyond the optimum point of the enzyme would rapidly lose its activity and will start to permanently denature, making the enzyme unable to feed the substrate. Basically, the shape of the active site would be changed if the temperature increases more than what is necessary. On the other hand, when the temperature is decreased, enzyme activity reduces as kinetic energy of particles is less, resulting in slower molecular motion and less frequency of successful collisions among particles. Secondly, let's talk about the effects of pH on enzymes. pH is the measure of the degree of acidity or alkalinity. I hope you all know about the pH scale, which ranges from 0 to 14, where 0 to 6 is acidic, 7 is neutral, and 8 to 14 is alkaline or basic in nature. Most enzymes in mammals' bodies work best at pH value 7, but there are exceptions like pepsine, which has an optimum pH of 1.5, and lipase, which has an optimum pH of 8. Unlike with temperature, if the pH value increases or decreases beyond the optimum value, a sharp decline in enzyme activity is seen, but generally these effects are reversible when it comes to pH. In summary, the 2 main factors that affect enzymes are temperature and pH. If the temperature increases beyond the optimum value, the enzyme would denature, losing its shape and unable to catalyze the reaction any further. In an increase in pH or decrease in pH, the enzyme would lose its activity, but only temporarily. Finally, I will be explaining to you guys about 4 examples of enzymes. Firstly, catalase enzyme, which is a common enzyme found in every living organism exposed to oxygen. Purpose of this enzyme is to catalyze the decomposition of hydrogen peroxide into water and oxygen. All fruits contain catalase enzyme, but some fruits have more. For instance, kiwis, Peaches, bananas, cherries, watermelons, pineapples have more, whereas other fruits like grapes and apples have low amounts of catalyst. Remember that the yeast fungus too has this catalase enzyme. Secondly, bromelain enzyme, which is a proteolytic enzyme or an enzyme used to digest proteins found in fresh pineapples. This enzyme is used to treat muscle injuries as a digestive aid, as well as this enzyme is a natural anticoagulant that works by breaking down the blood clotting fibrin. And it digests proteins, therefore it is also used in cooking to soften tissues in meat. Thirdly, we have the papain enzyme, which is again a proteolytic enzyme extracted from the papaya plant. And finally, amylase enzyme, which is a group of enzymes that hydrolyze or break down starch. Amylase is generally secreted from the pancreas or the salivary glands and has an optimum pH of 7 and an optimum temperature of 32 to 37 degrees Celsius. In summary, I talked about four main enzymes. Catalase enzyme used in the decomposition of hydrogen peroxide into water and oxygen, found in many fruits and almost all living organisms exposed to oxygen. Bromelain enzyme, a proteolytic enzyme found in fresh pineapples, used to treat muscle injuries as a digestive aid and in cooking to soften tissues in meat. Then the papain enzyme, proteolytic enzyme found from the papaya plant, and amylase enzyme secreted in pancreas and salivary glands that hydrolyze or break down starch during digestion. Lastly, I have some additional information. Their proteolytic enzymes are those enzymes which help to break down proteins into smaller fragments called polypeptides, peptides, and amino acids. Enzymes that work best in acidic conditions are renin and pepsin, which are found in the stomach. Enzymes that work best in alkaline conditions are intestinal enzymes, and the rest of the enzymes found generally work in neutral conditions. Also have in mind about the hydrogen carbonate indicator, which is a pH indicator harmless to plants, and it turns red in neutral or alkaline solutions and yellow in acidic solutions. So we have come to the end of this unit. Hope you all understood everything. If you all have any more doubts with regard to this lesson, please put them in the comment section below. Thank you all for watching. And if you all found out this video to be helpful, please do not forget to like, comment, and subscribe to my YouTube channel. It would be a great help. Thank you and have a pleasant day.